Yes, well, thank you very much for the introduction. I'm very happy to be here today and to give you some insights into our center and the Senckenberg Institute. And like I said before, we are looking at particles that are left in the environment and that we can analyze. The air, that's fleeting, that is a difficult area to do research in, but we're at the beginning of it. And in my presentation today, I will talk about the soil and the water, where we can analyze these traces to get a better understanding of our environmental conditions and also the ecosystems that live in our world. But first of all, I'd like to explain the basics. For us biologists and natural scientists, this is something that is clear to us. But biodiversity, what does it actually mean? You know, insects dying out, that's something that everybody's familiar with. But diversity you have in many fields. You have the ecosystems, those are the areas that we are in. These are very dynamic areas which are interacting with the inanimate surroundings. And then you have the species. We describe different types of species, of animals, or plants. And what is important is that these species are not only organisms that you have in situ, but that they have their function. And the community that they live in are functional communities. Functional communities that provide certain services for man too. But at the basic of all that, of the diversity, you have the genomic variation. So in other words, the change, the modification, what is in our genetic code and makes us different from other individuals. And this individual genetic code is the basis for the biodiversity that we have in the world. If you look at what is ecosystematic diversity, take a look. This is a picture taken in Austria in the alpine lands, and you see the forest interacting, cultural environments, you have valleys, mountains in the background, and each of these habitats together form a mosaic. And I wanted to use this image of a mosaic to show how manifold these ecosystems are and interact. What is the diversity of species? Well, that's what we know in terms of organisms. If you have different plants in your garden, then you have different species, maybe in different variations. But it's the different organisms that we are familiar with and that we appreciate. My favorite bit is special types of flies. They have their larvae underwater, and like small moths, they leave the aquatic environment, and then these cooker flies actually live then in the bushes. And there are 16,000 types of cooker flies the world over. And the special thing about this fly is that you find that in any type of water except for the Antarctica. And there, the caddis fly fulfills different functions. Some of them are predators, others eat plants, and they have completely different functions. And this diversity, functional diversity, and the variations that they build, this is something that I would like to show you on the right-hand side. So what is genetic diversity? The basis for it all. This is an example of our own research where we looked at how different these communities are moth in Papua New Guinea. What you see here on the left-hand side is a map, and on the right side you have the types and their genetic diversity. So each part is a genetic variety, and there you see the type of variants and how different they are, and it shows you from which part of these forests these variants come. So you have here this type of moth, and in this network you see where the individual variants in the individual sites occur, and these variants are very, very different. In the center you have a situation where we have all types of variants that are very far removed from each other, but they occur in different locations. And then on the left side of these three networks, you have all different variants are very close to each other. And these variants of the caddisfly do occur at all of these locations. So this is the basis of biological diversity. And how do we get there? Mr. Leber already explained it very nicely, but we basically read the genetic code of these individuals. So you have different individuals in a table, 4 different types, some with only one individual, others with many individuals. And there's different types that are being conserved. And some of these characteristics are the same. But there's some areas where we do have variations. And this genetic diversity is exactly what we're looking for. So there are species in between. So in themselves they are the same, but they are different from other species. But then we have individual cases where we have variations within the species. And that is what we as bioscientists read when we talk of biological diversity. Why is this important? It's important to understand why DNA as the carrier of this information is such an important tool for us, and why the environmental DNA is such an innovative and fantastic technology to understand these ecosystems better. We all know the DNA is the carrier of species, diversity of species. You have as a double strand ATGC that covers all variations of life. This is this computer code that Dr. Leber mentioned that is being carried there, and you have present in each cell. But this DNA is not only in our cells, it's also everywhere in the environment, in all parts of the environment. Here's a small example of a shark with its excrements. So you have DNA molecules from the excrements. So we can take a water sample, and then we will catch these molecules from the excrements of the shark. Simple it sounds, but it's only been possible that once we have been able to sequence these DNA strands. So what does it look like? Organisms, they will have a waste of excrements or scales, and in there you have DNA particles, not the entire strands. They are too long, they're not so stable, but the smaller fragments will come out of that, and they will then bind with certain particles or are completely free as a medium, in a medium like water. And this environmental DNA and we also call it eDNA. And these fragments that you see at the bottom are very, very small. So it's not these basic base pairs that we see, but only small fragments. And we need to see these fragments and then put it together in a biogenetic way to get sensible information out of it. So this is only a part of an organism that has been in the organism at one point. So here you see the rate from a bullfrog in the way it is reduced, but even after 60 days, the reduction rate, after 60 days after a bullfrog has been removed, the degradation rate is there, but even after 60 days, we could still show that it has been in there. So it is a technology which is and this is a completely new possibility for us in genetic research to be able to still prove the presence of DNA. So we as university scientists get the task to analyze certain environmental communities. So there was a chemical accident in some body of water and we want to know what happened there. So the organisms that you have there will be caught, will be analyzed optically. So we have the taxonomic experts, and then that information gives us the information on what kind of community it was in. So this is quite a difficult task, but there are other methods to obtain similar results. We have the metabar coding. So we put all of these animals into one blender, and the DNA is extracted from those three, and then we use PCR. All of you are familiar with PCR tests. Since pandemic, PCR is a word that everybody knows, but PCR is a very, very central tool for us, and is used here too. So with the PCR technology, we can take a certain segment of the genome that we then multiply, expand, and then we can analyze it and see what kind of cohabitation it's in. And the environmental DNA makes it possible for us to go one step further. So from the water, from the medium, the ground, the soil, or the air, we can extract DNA through a filtration, amplify it, and then go through the same sequencing processes to see what kind of symbiotic community has been formed. And it's faster and it has certain advantages, but why do we do that? Well, here you have an example from a frog in the water. Just imagine you are in the rainforest in Bolivia and have to do an analysis what kind of frogs occur in this body of water. So they do it by sight or acoustically. They listen to the sounds. Sounds easy. But once you're in that rainforest in Bolivia, you have a lot of sounds. It's incredibly diversitous. So try to get up at 5 o'clock in the morning, you walk through the bush, and you hear 50, 60 different kind of birds. And the same is in the rainforest. You know, to simply distill this to a specific type of frog, it's very, very difficult. But going through this analysis of the eDNA by analyzing a drop of water, you can see what frogs have been in there. So what is it that the experts can pick up with sight and audio, and what can we pick up with DNA? Well, what do? We go into the country. We are out in the great outdoors. That's a fantastic privilege of our work, and we take water samples. We filter these water samples, and from the filters we extract DNA, and this genetic information is being sequenced, it's being analyzed, and then we can allocate it to different species. And then we see the symbiotic community of different types of frogs. If we do it in such a way, we get to the following result. So there are 25 species that have been proven through eDNA. Now the experts, we were also able to prove 25 species, but interestingly enough, only 19 have been found by both groups. So the reason for that is that there are certain frogs that live in the trees. And so by doing water samples, they will not prove in the eDNA. So there are 6 types that can only be proven acoustically. But then again, there are 6 species that were not being observed, but that they did leave traces in the water. So you see, the tracking rate is slightly higher with eDNA, but of course we have to be knowledgeable of the fact that if a frog lives in a tree, we won't be able to trace it in the water. But if you have groups that have a major diversity, then of course eDNA is a lot easier to do than sending a group of experts in there to listen to the sounds. So with the environmental DNA, you can have a better monitoring of those habitations. So this type can be analyzed much better. If you look at our pyramid of biodiversity, we are here at the top triangle, but we don't look at the genetic variation. So we only see what kind of species occur simultaneously. In the next example, I would like to talk about a different type of application. So it can be used as an early warning system for pathogens. This is a crayfish plague. It's a very invasive type, the worst that we have in Europe. And this plague came through American crabs, and they have a 100% mortality rate. So if you have that fungus in the water, the local crab will die. And you can detect that because the crayfish plague leaves messages and traces, and these traces are waterborne. So by the multiplying technology and the eDNA, we can take it from the water and the tissue.
So the existence of the crayfish plague is easy. So since it is established since 2009, this system, the detection, and we have got the monitoring program to protect this crab against this plague. And the colleagues from Senckenberg, Gelnhausen, so for the environmental protection, they have a warning system for these pathogens. They did a study, a pilot study in Hesse. It's near the region of Frankfurt. You maybe know that region. And through probes and visual probes, and with the eDNA, they try to analyze the animals. And what was shown that the visual monitoring of these animals in these 4 points is depending on the activity of the animals. If the animals move a lot, of course, they lose more cells, and the omicedes that are distributed stronger. So with the activity of the animals, we can represent, especially in the warm months of the year 2030, a higher detection capacity through the eDNA. If the activity of the animal is lowering, we can recognize that also the capacity of detection is lower. The same is valid if I'm inside the effluent and try to identify those animals visually. The more they are active, the more I can catch, of course. And the advantage of the eDNA is in this representation. What we have is on the lower level the sample points, and then from the sample points A until H, these are the violet circles. These are detection points and results. You can see this scale here, which is logarithmical. You can see already in the point B, you see that there is a crayfish plaque. If I have a look at the diagram, then you see the results per time and per visual inspection. If the invasive crabs are there or if the crayfish plaque is in place. And the detection is not logarithm, and it's represented in orange. This means that I can recognize earlier with the eDNA if there is a crayfish plaque in place or not. And this detection system can be transferred to protection concepts. The eDNA and the pathology are both the eDNA is, of course, helping and directly correlated and linked to the population. If I have got a higher population, of course, I can detect it earlier. So this information could be used to idea protection plans. So on the left side, we can see in the Mümling, in the affluents, there is no plague, so still we have got healthy crab population, and lower down we have got the plague, and so the concept, the idea is that we have got here a limitation of this plague. If we have, for instance, these both methods, just a visual detection with the eDNA and compare both methods, then it is pretty clear that the eDNA has a lot of more advantages. We can detect the whole year, and always if there is some activity, and of course we need less samples to have a certain monitoring. And the monitoring about activities are more sensible than we will do it through visual detection. So in this manner, we are having a closer look to the eDNA. It has more advantage to the conventional method. It could be applied perfectly well to detect invasive species and to avoid migration of these species and to optimize it for the detection of certain species. So we are working on several levels, and it is important to know which individuals are resistant to this pathology. And the third example, and we are talking about the beech and the beech leaves. So having a look at the beech leaves, we can see all the community of all insects and detect all insects in the forest. This was a really surprising fact for me, but it has sense because the animals that are in the beeches and then the leaves of beeches, they leave, of course, eDNA. These traces are due to, well, maybe feces or the production of feed. And through the leaves, we have got these DNA traces from these animals that has been there or are still there at those leaves. And we have thought about that. How can we analyze it deeper? And again, I go to the bank of samples from the federal states. It is a data bank that was established in the 80s, and through biology samples we can trace our biotical changes. And since some years we are talking about that we try not only to analyze the contaminants, also we try to analyze the biology changes. And the great thing about that samples is that for these samples we have just a couple of samples, but there are thousands of leaves. So these thousands of leaves, we take them from one tree and they are frozen directly and they are milled and analyzed. And I have got a very good reproduction of the eDNA. And these are samples that could be traced 30 years ago even. And for a long term detection, it is a very good option because we have got conserved very well those samples. So what we have done with those samples, we took the samples and extract the eDNA and sequence it and could have a closer look to the biological community. And so we could see which species were at the forest. And the different species of trees had different insect communities. We could see, we could appreciate different types of nutrition species depending on nutrition. So we could analyze that really well in comparison to an expert who observes the forest. And just through the community of animals, we could recognize which type of tree was sampled. We have got a very strong link between the different trees and the population, animal population. Every species of tree has a known insect community. And this was very interesting, but not enough. We were very interested in the changes. What has happened below the species? Of course, we can see which species were in which tree, but what happens to the genetic variants? They are the base for any change. And if we have a closer look to two examples, we have got here wasp that is living on beech and on the poplar tree. So we have got different variants, species variants, and what we could recognize is that we have got specific genetic variants in the beech and some on the poplar tree. So we have got really an ecological difference in between a species, and we could trace them in a genetic variation. Also interesting is that we have got also time-lapse dynamic. We have got an animal that has two different variants, and we know it since the beginning of 2000s. And in both variants, we represented in red and blue. It's a very high density of population. And the blue variant, well, left, and the red is still there. So this means that with this eDNA, we can not only recognize which type of species are in place and also how they are distributed. And this is evolution. Evolution that's just taking place in a local level, because we recognize that the population in the different genetic variants is popping up in certain sites. So we have got arthropods that are leaving traces, and we could differentiate. So, and then we have got, of course, for every year, a new period. We set them on 0 every year, and then we have got also a data store for long-term observation. And then we are moving on 3 levels. Just to give you a last example, we are talking about changes of the that we can have a closer look at them in the sediment. Also, the sediment cores are still for high value for us. We have, for instance, the Lake Scharmützel. We have got a sample that started in 1890 up to 2010, and we can see the change of the landscape. So we have got also in this time, we can see that we have got different situations. We have got at the right time, up to 30 years ago, we see that the community has changed totally, and then from this point on, we had got a growth, at least again to an area which is different from the left side, from the original situation, but it is again going to the original situation 150 years ago. That means that 30 years ago, when we developed the new installation, water installation, we have got improvement. So with the eDNA, we can really visualize different species. We can observe hundreds of samples in a parallel manner. We can do that in the sea, in the air, in the soil. It's cheaper than an expert analyze, and it is standardized. But nonetheless, we don't have a lot of standards, and still we need highly technical expert knowledge, because there could be contamination taking place, and we need experts to support us. So with it eDNA, we can investigate the eDNA, we can investigate the landscape and do some forecast. See that the different parts of that the different influences could affect the environment. So thanks a lot for your attention. Thanks a lot. Please come to us. I have the first question. What means the eDNA through soil, water, and for the human body and for the human organism? That's a very good question. The DNA per se is inert. This has mentioned already Mr. Lieber. It's the code that is read by the computer. We have got information that is inactive in these traces. In a functional cell, it will be read to generate protein. That means that the only thing that could happen, that organism would absorb these DNA and cooperate, but the human body per se is not doing that. We are not very worried about that. What do you notice when you investigate? How is the nature changing? We are all talking about the climate change, and we are talking about sustainability. Even in the financial industry, it's a huge topic. What is happening right now? For us, for our long-term investigation, it is clearly that the change of the landscape and the change of the habitats and the components of the habitats, of the variety of these habitats, that has an effect on the variety of species. The more variability we have, the more species we have. That's pretty clear. But what's happening in Germany with the insects in Germany, the insects dying, the data are very clear. There is an area of the soil investigation that we are losing species in the forest, for instance. We know this tendency, and we can see these tendencies also in we see in the water. So we don't see only not a very high change in the species. So we know that there are less species in place. So what's the timeline you're looking at? So we hear now that the water in Venezia is now very clear. Is it a long-term vision? So we noticed it already. Yes, it's not a scientific study we are evaluating, but we notice it clearly. Maybe you notice it, for instance, in the last year. From my point of view, we had a very good insect year. We had a lot of insects at the springtime, and the summer was very good. And this year seems to be the same. 3 years ago, it was a bad year. So the insects react. They react on the environmental conditions of the last year. So this means there's a high dynamic, and this is very difficult just looking at just in a few years and see the tendency for a long term. What we still miss, long-term studies, and this analysis of the sample bank. We can have a look 30 years ago, where a lot of happens in Germany, and we are working on that with pilot projects to have a further look and to trace this question from an old treatment plant. So you can have a look. You would have a look. It's different sample from different years. You could really have a look at everything. Yes, you can do that through the eDNA. We have got these studies in the Goethe University. I didn't participate on that, where you could trace the coronavirus through the treatment plants.
All right. The wastewater from the cleaning plants would be an El Dorado for any scientist, absolutely. I never thought about these sewage treatment plants as an El Dorado. Well, it's all being filtered, don't worry, everything is nice. Okay, and preventively, if you were talking about an early warning system of pathogens, what can we imagine for people? Well, these are all good systems for early warning systems. If you think of people and food intake. So if you have invasive species that will attack our vegetable resources or our animal resources or the fish, then of course it's good that we can react at an early stage, and the earlier the better. And fishery, that's a huge industry in Europe, and of course they're invasive species. That's a terrible thing. So And many of these species, in fact, come through the rivers. I mean, that crab, is being used to be eaten by many poor people in the past instead of fish. Now it's considered something very special, but back then it was actually poor people's nourishment. And now, because of the invasion of the crab plague, you have a direct impact on human development. And if we recognize such systems at an early stage, that's always good. Okay, just for the last few minutes, a detailed question. It's summertime now. What kind of services for human beings are performed by wasps? Well, wasps, they are very good at foraging on dead animals. If not, we didn't have wasps, we would have to have the cross taking care of that job. And wasps, pigeons, and cross, they are very, very good. The pigeons too. Yeah, but the pigeons, they create waste, but they also eat a lot of waste. So this is an investment conference now. Ever been in touch with the industry, or are you under the radar? Well, there's a couple of startups around eDNA, and at our Löwe Center for eDNA, we are in that transnational transfer. So we have stakeholders, and we have those that we work with. At the moment, I have to tell you the truth, we're happy if we have additional parties that are interested in us, but because at the moment, the industrial corporations that we have are with companies that are in the field of sequencing and use us to develop new protocols just for the genome sequencing and interpretation. Our products are not so hot for them. So we're not at the point yet that the industry is reaching out to us. So when you talk to Jinsky, who comes from the Fraunhofer Institute, we do fundamental research, of course, and those that do applied research, you will see that there's more interest. Yeah, but we have a cooperation with them at the Löwe Center, and really, for fundamental research, which is then passed on through the Fraunhofer Institute, the transfer points, and the stakeholders. So basically, the entire process is being covered. All right, so what we learn is what we heard from Dr. Weber. You can confirm that. Small, cheap, fast, and looking into the future. So the Senckenberg Museum is opening its doors. We can talk about that too. Go there, visit the Senckenberg Museum in Frankfurt. Yes, we would be happy if people came to our department too.